have some chapter 3 left, but that's okay. But I do think I'll finish covering all chapter 3 before the first midterm. You okay so far? So we've been talking about the Metazoan's dilemma, which is how do you make thousands of different kinds of cells, each of which will do its job and be in the right place in the entire animal in order for the animal to function and, most importantly, for natural selection, reproduce, right? Because that's all natural selection cares about. Not is it elegant, not is it pretty, but will it produce offspring? And we looked at Sidney Brenner's pet animal, pet Metazoan, C. elegans, and we saw that there's this relationship between the mitotic lineage of every cell and which fate it takes on. And you might ask, well, is that a coincidence? I mean, maybe cell specification has nothing to do with mitotic lineage, right? Maybe it's to do with factors that are out there or something like that, and it just happens to correlate perfectly with mitotic lineage. Everyone see that? But there are all sorts of reasons to think that's not the case. So, for example, eventually people worked out the sequence of gene expression that decides fate. And what they found is messing about with any of these genes would prevent this particular cell from becoming a touch receptor, for example. And another thing that indicated that it wasn't just a coincidence was that when we talk later in chapter 7, we'll talk about cell death, which is a natural part of development in all plants and animals on Earth and has been for a long time. Okay, some cells get made and then they never quite take on a function; they just die, right? And in some of these lineages, when you come out to the last little final division, in some cases there'll be a final division, and one of the cells will die, and the other will take on a function. And, so researchers ask, well, I mean, is that cell that's going to die, is it really committed to die? Can we change its fate? And so what they could do is use a laser, put a laser through the microscope, kill the cell that was going to become a functional cell, and then see what happens. What does the other cell do? Does it die, or does it take over the fate of the cell that just got killed? And the answer is almost always it would have died anyway. And the animal had to do without that particular cell. And that's pretty mysterious, right? Because up until that last division, you have to think the cytoplasmic factors inside the mother cell that was going to go through her last division, you have to think that they got it divided pretty evenly between the two. And yet one is fated to die and the other's fated to live. And so what that tells you is that this fractionation of transcription factors is very, very precise. And it also tells you, or the other thing I want to emphasize, it's not just which transcription factor happened at this division, but how the transcription factors got divvied up in all the previous divisions in the mitotic lineage. And unfortunately, it's not static in time, because as the cells are dividing, one set of transcription factors may very well turn on the expression of another set of transcription factors. And so each cell has its own unique history of exposure of cytoplasmic factors, various transcription factors that make it unique and therefore give it its unique fate out of the 1,000 cells in this worm. And questions about that. And early on, there was lots of talk in the late 20th century, about the time I'm a professor, about cell fate and cell commitment. Is the cell committed to its fate? And in the experiments I just explained to you, they seem to be pretty committed, because even though you kill its sister cell there, it goes ahead and dies. So, but I do want to point out to you that the term, to say a cell is committed, to say it's committed has no meaning unless you specify committed in the face of which challenges? So that cell, we killed its neighbor that was going to take on a function, and it goes ahead and dies. But so in terms of that challenge, it sticks to its fate. I'm giving up the ghost. I'm done. But, with enough information and power, I could go into that cell before it dies and fiddle with its gene expression and get it to take on another fate, right? So commitment, and we'll run into this again shortly when we talk about other animals and C. elegans. When we say that a cell is committed to a fate, it's always relative. And here's an example that maybe help you'll think of it. I feel like my wife's committed, right? And so far, things are going well. And she is, and I don't doubt that. But that's in the face of the challenges she has today, right? If tomorrow she meets a tech billionaire who says, would you like to have an island? She may decide her commitment to me is, not so ironclad after all, right? And who could blame her, right? So I'll emphasize, and I have to say, in the late 20th centuries, people were talking about these things in these studies. There was a lack of appreciation for the fact that to say a cell's committed has no meaning unless you specify what challenges it stays committed in the face of. Okay? So that's pretty cool, that you can divvy up transcription factors and end up with 1,000 different cells, more or less, each of which has a different fate, a different function, a different structure, and is in a different part of the animal. And you know what? Whatever you think about that worm, it's good at making more worms, right? So it works. The only thing evolution cares about. And the question is something like that happening in other animals that have more than 1,000 cells? And I'll telegraph the answer. No, that's not how it happens. To talk about that, we got to talk about embryonic development in other species. So in a frog, say, the fertilized egg, the zygote, goes through a certain number of divisions, and at first they're quite symmetrical. And so there is a power of 2 in the number of cells total. And eventually you end up with a blob of cells that's more or less spherical. And from here to here, we start calling this an embryo. And when it reaches a point where it's a blob of cells, pretty soon what happens is the embryo becomes a hollow sphere of cells. And we have terminology they need to know for that. We call it a blastula. And the cells that make up the blastula are called blastoderms. We'll run into that again. The derm kind of gives the point that it's on the outside. And there's a hollow fluid-filled space inside called the blastocele. And as the blastula continues to develop, the blastocele will get smaller and smaller and smaller until eventually it disappears as the cells continue to grow and squeeze that cavity closed. And what happens is, at one point in the blastula, a little dimple appears at one place. And the dimple happens to be located, by the way, where the sperm penetrated the egg. I wonder why it gets penetrated there. Oh, there's a structure in the egg that's there already, as we talked about before. And there's an inhomogeneity inside the egg. And what happens is, once it starts developing this dimple, it starts growing invaginating, and will eventually go all the way through the blastula, right? So now there's a tube. And at this point, that tube, that's the beginnings of the GI tract. So you want to make an organism, first thing you got to make is a gut, so you can take in nutrients. And as we pointed out earlier, in some animals, the place where the dimple first forms is going to form the mouth, like in jellyfish and the like. And in other animals like us, this dimple forms at the end that's going to end up being the anus. And at this point, because of his invagination, now the embryo is divided into 3 layers. And I'll remind, and the layers are called the ectoderm, the endoderm, and the mesoderm, mes in the middle. And I'll remind you that this way of making an organism has been around a long time, because remember back here, it's after the Cnidarians. As soon as you get in all the other animals, they are all making 3 embryonic layers: ectoderm, endoderm, mesoderm. The details of how the 3 layers get formed differs in different species, and is yet different in mammals, but that's the basis of it. And the next thing, it's been a blastula, and now it's been a gastrula. And the next thing every organism makes is a nervous system. So first you got to have a gut, and then you got to have a nervous system. And that shows you how preeminent those are in terms of what it takes to make an organism. And at this point, once it starts making a nervous system, we call it a neurula, and that process of making a nervous system is neurulation. All this stuff you might have heard before. Hope so. And then for frogs that undergo metamorphosis, they hatch in the larval stage, and then there'll be metamorphosis, and the adult will make gametes and start the whole thing over again. Questions about that so far? Yeah, good.
Where the sperm merges with the egg?
Where the dimple is? Yep.
Or merges with the egg, correct?
Yes.
So the egg is not, you stay all around.
No, it's not.
So does that mean that the sperm recognize where do you just stay out?
Or that the egg will only let it penetrate in one place. I mean, one area.
But
good. Yeah. See, I'm going to say the wrong one. Tell me your name. Justin. Okay, that was right. Okay.
If it's many cells, how do you know where sperm?
So they could, yeah.
How did they work that out? I'm not going to talk about it. It was complicated. And I don't think it was in frogs. Might have been in sea urchins, where gastrulation is different. And yeah, Laura.
You said that the egg might have a mutation that it doesn't like this stuff. That is that was good. Like,
yeah, it's a great question, but given that we said there are inhomogeneities inside the egg cytoplasm, it's probably crucial for there to be one place where this gets started in order to determine the planes of cleavage, and therefore to determine how those things get divvied up. And so mom might make an egg that doesn't have that sort of characteristic to it, but if it gets fertilized at all, it's not going to work. Abner.
Hey, insane or other.
So far it's mosaic, yeah. But first let's talk about what the 3 germ cell layers do. Those 3 germ cell layers are going to make up the entire body of the individual. There might be other cells, as we'll see, that don't contribute to the final product, but they're going to make the body of the animal. The nervous system comes from the ectoderm, the outermost layer. And it might make sense to you that the outside of your body, that's where you sense the world, right? So it makes sense that if you're going to have touch receptors and things like that in the exterior of your body, that it'll be the ectoderm that's going to make the nervous system. And as we'll see in a moment, much of that nervous system will then embed itself inside the organism to be nice and hidden and tucked away. And so it's going to make the nervous system and the outer surface of the skin, the epidermis. So what we're going to see later is that for these cells, there's a push-pull going on where they're going to take on one fate or the other. They're either going to become epidermis or they're going to become nervous system. And we'll talk in detail about how they make that decision. That's a fundamental decision that ectodermal cells have to make. We won't talk much about the derivatives of the mesoderm, making all those middle layers. The ones we'll talk about the most are the muscle cell. But it also makes, bone tissue. And we will talk about this mysterious structure, the notochord, short. It's mesodermal. And then the endoderm is going to make all the guts, all the stuff inside. And I'll barely mention any of those things again in this class. Be different if this is developmental biology, but it's not. Got that so far? So, thought I had another. Okay. So, in mammals, things are a little bit different. In mammals, when the blastula implants in the endometrium of the uterus, it will start taking a very complicated structure that we haven't seen before. And if we take a look here, we'll see it's like two fluid-filled spheres. And on the undersurface of the smaller one, there is what forms is the inner cell mass. And so in mammals, the entire body is going to get made up of the inner cell mass, and all these other cells, they're there to provide the support system for the fetus, the embryo and fetus, to continue to grow inside mom. Totally strange thing, but then all those mammals do it. So gastrulation is different in mammals because of this. In mammals, gastrulation, the making of the three layers, happens when in the inner cell mass, the smaller portion there, a primitive groove, primitive streak arises. Just a little groove in the mass of cells there. And if we look at it in cross-section, what happens, and by the way, the head is eventually going to form at the node, the beginning of the groove. And what's going to happen is these cells are going to start migrating in a complicated pattern towards the center. So imagine these cells, and they're moving and growing, and they're migrating in and eventually dimpling out again. And so they will eventually make three layers, because the first ones that dimple in are going to make the endoderm, and the later ones that come in are going to make the mesoderm, and some are going to stay on the outside, and they're going to make ectoderm. And we care about them because they're going to make the nervous system next. How's that? So far, all the questions have been great. So keep them coming. So here then is another view of the primitive streak and what's happening. And the groove gets deeper, and we put a new name on it. We call it the neural plate. It's just this relatively flat groove, and it's going to make the nervous system, so we call it the neural plate. And it's made of ectoderm, of course. And what happens now is, because of cell movement and cell divisions that are uneven, it starts deepening that fissure to the extent that eventually the tops of these two sides of the groove will meet in the middle and merge. And then what happens is there's a tube of cells that are now below the surface of the organism. And there were also some cells that were at the very peak of the groove that will break off as well. And this tube we call the neural tube, and it's going to make the central nervous system, the brain and the spinal. And this hollow space in the middle of it, in the brain, as the brain gets more elaborate, it's going to make the ventricles. And as you know, those ventricles connect to a tube that goes down the center of the spinal cord called the central canal. So the structures are foreshadowed already. The rest of the nervous system, the peripheral nervous system, is going to derive from these cells called the neural crest. And as we'll see in a couple of chapters, these neural crest cells are eventually going to migrate out throughout the body and make the entire peripheral nervous system, autonomic nervous system, enteric nervous system, etc. How's that? The neural tube forms first towards the head end of the organism, but not all the way at the front. So the tube forms here, and then as development continues, it's like, imagine a Ziploc bag where that part that's put it together in the middle and then squeeze out in both directions. And so the tube's going to form and close up in both directions. And eventually, at the head at the end, where the primitive streak began, the node, that will develop much more divisions, etc., more cells getting made, and it's going to form the brain. And pretty early in development, you can see several divisions of this neural tube that are going to form the various divisions of the brain. So the foremost portion is going to make the forebrain, also known as a prosencephalon, which consists of a telencephalon, that's going to make the cortex, basal ganglia, stuff like that, and the diencephalon, that's going to make the thalamus and the hypothalamus, structures you've heard. Another portion, and they all look like kind of bumps around here, is going to make the midbrain, also known as the mesencephalon, and another is going to make the hindbrain, also known as a rhombencephalon. So it's the hindbrain that's going to make the pons and medulla. All things you've heard about before. Laura?
They all made up the same type of cell.
And they're all made up of the same type of cell. And so I know what you're wondering. Are they already specified somewhat differently from one another? And you can't ask that yet, because I'm going to tell you in a bit. I see where you're going. Okay. And then the rest won't grow nearly as much, but will form the spinal cord. So we can now look at various portions of the ectoderm. I already told you that many epidermal cells are going to make the epidermis, and they will also make various structures that we'll run into in the future, like the olfactory epithelium, the mouth epithelium, the lens, and the cornea, all of which are important for the nervous system to work properly. Others, the neural crest cells, are going to make all the peripheral nervous system, as well as a few other places, like the adrenal medulla, the inside of the adrenal gland, melanocytes, they're going to provide pigment, facial cartilage, that is important for forming a face, and dentine for teeth. The neural tube is going to make the central nervous system, including all these things. Brain, spinal cord, etc. Oh, but it also makes the posterior pituitary. That's important. Pituitary, I'll remind you, consists of two parts, right? And one part is derived from the nervous system, and the other part isn't. More about that in chapter 7.5. So, the question Laura was trying to get at is, are these embryos, are they like C. elegans, where there is the Mosaic specification of cell fate, and or that mitotic lineage specifies cell fate? And the answer is no. And in the early days, it sort of looked like maybe it would. One of the earliest experiments was when someone looked at I think a frog embryo or fish, I forget. And he made a very fine red hot needle, and at the 2-cell stage, he popped one of the cells and it died. And after that, the remaining cell, when it kept dividing, etc., made half an embryo, like somebody had split an embryo in the middle of the sagittal plane. And so that sort of thought, well, gee, maybe even at the 2-cell stage, this, cell fate is specified. But there's a flaw with the experiment. One is, what happens when a cell continues to divide next to a cell that's dead? Necrotic tissue, toxins, who knows what's going on? So a later experiment by Hans Dreich looked at hydra, like primitive animals, freshwater hydra, they're everywhere. Anywhere you see pond water, there's loads of hydra out there. So they're easy to get. And with a microscope, he waited until the hydra was at the 4-cell stage, and he teased the 4 cells apart. So is each one going to make a quadrant of the hydra? The answer is no, that's not what he saw. Each one made a hydra. Each one was smaller, like they had less starting material, cytoplasm, to make one. So instead of making one normal-sized hydra, it made 4 small ones. And that suggested that the fate of these cells was not fixed, that this was not a matter of Mosaic specification of cell fate. And eventually, a German scientist, Hans Spemann, did one better by doing an experiment similar in vertebrates that also suggested that cell fate could be changed, that there was not a Mosaic specification of cell fate. He took a human hair and made a little knot, slip knot in it, and then got it into the microscope where you could see, and then used his tweezers to pull the ends of the hair until it came in between the 2 cells in a 2-cell embryo. Everyone get the idea? And it turned out, he failed the experiment a whole lot when he had an idea. Wait a minute. His infant daughter, her hair's a lot finer than his. So he gathered some snippets of her hair, some of which, by the way, were found in his desk after he passed away. And he found that by using that and making a slip knot, he could divide the embryo, and as long as at least one nucleus was in both remaining halves, 2 different embryos would form. How's that so far? And this ended up being part of a whole series of experiments where people found, like in frog embryos, so there's the frog embryo, and if it's using Mosaic specification of cell fate, then if he takes out a chunk, he should end up with a frog that's got some pieces missing, right? Maybe the head, maybe a limb, depending on what that chunk was going to make, right? But every time he did that, the embryo came out whole. No matter which chunks he would lop off, a whole frog came out. Or a tadpole, I should say. And so people started seeing this a lot and realizing that there is regulation going on. The embryo can regulate, or sometimes it'll be called self-regulation, where you do damage to it, and if it's not too much damage, eventually it'll compensate for that. And this means, when this happens, this means that cells that were going to take on one fate must have changed their fate. Because otherwise, if those cells he took out, if they were going to make a particular structure, that structure should have been missing. But no matter which parts he took out, a whole animal came out. So this is known as self-regulation. And by the early 20th century, it was very firmly established and recognized. And so that meant, something complicated is going on here, that it can compensate for that. More importantly, it means it cannot be Mosaic specification of cell fate, right? Because if it was C. elegans, I don't know if I said this, in C. elegans, after that first division, where a P1 gets made, a cell they call P1 or a cell they call AB, if you take a laser and kill the P1 at that phase, the rest of the cells go ahead and divide, and they make all the stuff that was going to be in the front of the animal. Of course, the animal doesn't work, right? Because it doesn't have a back, but they seem pretty committed, right? The AB cells seem committed to make a front, and they won't regulate if you kill the P cell. So, yeah. So this suggests, And by the way, any animal of any size, regulation takes place. Embryonic regulation, self-regulation takes place. So this makes you think that maybe if you're going to have mitotic specification of cell fate, maybe that can only work if you have a limited number of cells total. That if you're going to start making an organism with billions or trillions of cells, there's no way with a mere 20,000 genes, if they were all transcription factors, there is no way you could divvy them up so that they had to take on a billion different fates, right? So a long time ago, after our common ancestor with C. elegans, our ancestors took a different pathway, a different strategy to develop. And that was, they weren't going to try to keep track of mitotic lineage. Rather, they were going to have the cells talk to each other. And if some cells ended up missing, other cells would fill in for them. And you can see how there's some advantage there, right? Accidents happen. And if some cells get killed, well, maybe the rest will be able to figure it out. We're going to talk about this at length, actually for the rest of the course. But keep in mind that for this to work, notice that this already hints that the cells must be talking to each other, because otherwise, how would they know, right? How do the cells that change their fate to make up for the lost cells, how do they know those cells were missing? How's that? And so this self-regulation is very much at odds with C. elegans. So in the other embryos, vertebrate embryos, for example, first of all, this is how conjoined twins can happen. Imagine, for example, a Spemann-like experiment where an early embryo gets divided and they get pushed far enough apart that each one makes a whole individual. And then if they're pushed very far apart, they make, monozygotic twins, identical twins. But if they don't get pushed apart enough, then they start interacting with one another and communicating with each other. And so that you end up with what you see in conjoined twins, where every cell in their body has neighbors that are totally appropriate for that kind of cell. Again, they got to be talking to each other. There's got to be a way that that's going on. And what we see in conjoined twins is, something happened. There was an accident, not the normal. This means that at some point in the embryo, every cell is capable of taking on any role. We say that it is totipotent. And the big excitement about stem cells and how they might revolutionize medicine these days is they seem to be able to take on any fate. If only we could figure out how to direct them to take on the fate we want, i.e. for, dopaminergic cells in the substantia nigra for Parkinson's, something. And sometimes we'll see there are cells that aren't totipotent, but they are pluripotent. They can take on lots of different roles, but there's a limit. And so those are the terms we use, pluripotent and multipotent. And as we'll see, as development proceeds, cells go from being totipotent to becoming pluripotent, and as development proceeds, possible fates for any given cell start to narrow down. So that we'll see, again in chapter 7, if we go to a chick embryo early enough and lop off a limb bud, the cell will regulate and the chick will come out with all of its limbs. Even though those cells were going to make a limb, and you lopped them off, and then the other cells were going to make the shank or the thigh, they change their fate and form a whole leg. But of course, there's a limit to how late in development you can do this. You can't take a chick just before it's going to hatch and lop off a leg then, and it won't compensate. Or in rats, right? Rats at birth, if you snip off one of their digits, they'll grow it again. But of course, you can't wait until it's a juvenile rat and snip off a digit. It won't regenerate anymore. So there's also this concept that as development proceeds, we become less and less, the cells become less and less flexible in their fate. And you could say they become less plastic, which is not unlike what you've learned about a nervous system, right? That your brain, when you were younger, it was much more plastic than it is today. And sorry, I'm the least plastic of all in this classroom, right? You're not going to get away. It'll happen. Questions? Good. Tell me your name. Yeah.
It's okay.
One more time, though. Pramika. Okay. Yeah.
So when you say the cells talk to each other, right? So I'm assuming at that point in like development, there's no like fully functional like nervous system. So they
No.
each other be like gap junctions or like how do they
We will cover this in chapter 2, 3, 4, 5, 6, 7, 8, 9, and 10. That and that's the whole trick of making a really complicated nervous system. They're always talking to each other. Even into adulthood, it's just, where synapses might come and synapses might go, but that only happens because they're talking. Eric.
One of the standards, like for certain processes stuff, like you said, you could just take some out. Like, do you know when that, is there standards based on, we know, okay, if that happens at this time, it'll then develop?
Yeah, I mean, I don't actually know the details, but I know that it's specific to the species and pardon?
Depend about the complexity of the species.
I don't know that it's as simple as that, right? I don't know. And I don't know any metric of, it's probably not something simple like, well, once you get to 1 million cells, you can take out 300, 3,000 and nobody will notice. I don't know. Don.
You see like a similar relationship between monozygotic twins as they had hydras to separate.
Absolutely. What's happening with monozygotic twins is it's the same as when Dresch took hydras apart, except with a placenta to nourish them, they're the same size. I mean, in general, birth weights of twins in general tend to be a little bit lighter than that of singletons, but certainly not half, right? So more compensation, more regulation. Laura?
So why doesn't the whole organism form when you kill a cell in the 2-cell stage pretty fast on the other one?
Oh, in the early experiment, probably what happened was that the remaining cell, it was distorted by having a dead cell next to it.
Well, why doesn't that happen in the extant?
Yeah, it's a great question. And it may depend on species and may depend on what you do exactly. Notice in those other experiments, there are no dead cells next to the surviving cells, right? When you separate the hydra, none of them have a dead cell next to them, right? And when you scoop some cells out of that frog embryo, notice dead cells there. Justin.
Do they influence cells that don't have the same DNA as them?
Well, in the normal case, they all have the same DNA because they all derive from the same. Are you thinking about fraternal twins or something?
Yeah, like are the Siamese or
conjoined? But yeah,
are they always twins? Like
it's a great question. I think it might be possible for dizygotic twins to occasionally merge, but I don't really know. I'm not sure the answer to that.
Like not so the others, like even amongst their adults cut their like.
Oh, yes, excellent. So what you're pointing out is in some amphibians, animals are able to regenerate limbs, and I would explain that to you, but I would have won the Nobel Prize already. Because nobody knows why those species can do it and others can't. I think it's time for a Kahoot. That's what I'm thinking.
Yeah.
Here's what we're going to talk about next about one of the most classic experiments in biology and in embryology and what it taught us, and basically how once the embryonic nervous system gets started, how cells make decisions about who's going to be a neuron and who's going to be a supporting player. Not everyone can be a star. Back to Hans Spemann with those fabulous eyebrows of his. He would eventually win the Nobel Prize in 1935 for his experiments in what came to be known as experimental embryology, where you would take embryos of various species and do manipulations, physical manipulations of them, and see what happened to development. And one of his first claims to fame was learning about the development of the eye in either frogs or fish, I forget. And remember I told you that it's a different part of the ectoderm that makes the lens of the eye. And he was looking at the developing nervous system in these animals, and he saw that early in development from that neural tube, what happened is two little outcroppings came out of the front and formed little depressions that he called the optic cup, because they were going to make the back of the eye, the retina. And so when you learn about visual processing, and you're told that a lot of visual processing is happening in your eye, in the retina, that's because it's basically an extension of your brain, right? Those are brain cells that came out there to get the information as quickly as possible so that you could possibly make sense of so much information. But what he noticed is the epithelial cells in front of the optic cup would branch off, bud off, and form the lens. And then the cells that got left behind after it branched off, they would start becoming transparent to form the cornea. All right, so all ectodermal in origin. And how's that? And so he was asking, well, how specified is the system? And so he would do experiments like this, where if he removed the optic cup or optic vesicle, he found that the question was, would the epithelial cells out there make a lens anyway? And the answer was no. They changed their fate. They were going to make a lens, and they didn't. A lens and a cornea. Or what about if he took out the optic vesicle and stuck some other cells in there? Could any old cells get the epithelial cells? Was it just the loss of touch or contact? Could any old cell stuck in there cause the epithelial cells to make a lens and cornea? And the answer was no. Well, all right, what if he takes the optic cup and moves it further down the animal's head? What happens then? And what he found was, when he put it down here, now cells that were going to make skin start making a lens and cornea. And they just so happens to be right there where he put that ectopic optic cup. Well, would any epithelial cells respond to the optic cup that way? Not quite. If you brought the optic cup down at the trunk of the developing animal, the epithelial cells there didn't seem to be able to listen to it. And they didn't form. He called this process of the optic cup instructing the epithelial cells to make a cornea and lens, he called it induction. It's like they're inducing the epithelial cells to change their fate to make what the eye cup needs to make a functional eye. So again, there's got to be communication going on. And he expected that the optic cup was releasing some chemical that communicated to the epithelial cells and told them, oh, there's an optic cup forming here. We better make a lens and cornea. And just about any epithelial cells in the head could respond to that call. So they're all pluripotent. They're all capable of making that. And not all epithelial cells were. So already these epithelial cells must be different than these epithelial cells. They've already gone further down that pathway I talked to you about, and they're not as plastic. They're not as capable of regulation. Questions about that? So Spemann thought, you know, this induction business is interesting because you could imagine development of the organism, the epigenesis that Aristotle described but couldn't explain, right? The evolving and increasingly complex structures that are evolving. You know, you could explain all that by cells inducing one another. And they're all talking to each other the right way, and they're inducing things, and, you know, you end up with a whole organism. But wait a minute. For that to work, they can't all be wildcatting, right? They can't, because otherwise you'd have 4 or 5 individuals getting made from each embryo, right? There's got to be some organizing feature that says this is where we start, and now we start reducing cell fate, and all the cells are going to talk to each other and branch out from there. And so he was wondering, how early in embryogenesis can you find something that looks different than the rest? And he had a graduate student in the lab, Hilda Mangold, and he gave her the project to look at this, and she started manipulating, messing around with the dorsal lip of the blastopore. What's the first thing you see in the embryo, right? The first structure you see is the blastopore that's going to begin gastrulation. So, okay, what happens if you take the dorsal lip of the blastopore from one animal, say a pigmented amphibian, and you graft it into a weird spot in another animal, say an unpigmented animal of the same species? And I should say, these sorts of manipulations, the way Spemann was able to do that is he would make his own, I mean, these structures are tiny, right? He would make his own instruments by taking glass rods, heating them up, and pulling them out and stretching them. And if you broke them off in the right way, he'd end up with these incredibly fine-tipped glass rods that he could use to slice off an optic cup and move it around. So he had Hilda he taught her how to do this, and she did experiments like this. And what she found was, when she successfully got it to graft in there, that there would be a secondary blastula, a secondary blastopore form, and 2 neural tubes, and eventually 2 embryos, conjoined, like conjoined twins, and yet the dorsal lip of the blastopore, because the cells were pigmented, you could see they were tiny. Almost all of this organism was induced to form from the other organism's cells. Everyone get that? So these cells had co-opted the other cells and formed a whole new organism. And so Mangold and Spemann proposed that the dorsal lip of the blastopore contained an organizer that was going to start the process of differentiation, start the process of epigenesis, and start making a gut and a nervous system. And you can see the beauty of the appeal of that right now. So there's just one part of the embryo, the dorsal lip of the blastopore, and that's the starting point, the anchor. And everybody else takes their cue from that. And if everybody's taking on the fate that's appropriate for what their neighbors are up to, voila, you end up with a whole organism. Abner.
Experiments on destroying the dorsal?
Pardon?
Did they do experiments on destroying the dorsal blastopore?
On disjoining it. Oh, destroying it. And yeah, probably, and I don't remember what happened. Probably the embryo stopped and, quit working. I mean, yeah. I mean, if you just remove the dorsal lip of the blastopore, the question is, would the remaining cells form a new dorsal lip of the blastopore? And I don't remember. Yeah. What about the nervous system? Let's see. The cells that she transplanted from the other pigmented animal, they were all confined to basically the spinal cord. So it's not that these cells divided a whole bunch of times and made a whole new organism that has the genotype of these cells. Rather, they commandeered cells from the other organism and used them to make its body. How's that? So that was done in 1924. Mangold never saw her paper in print. She had sent it off, but then there was a kitchen fire and she died in the fire. So she never knew that she never got to see her published paper. She knew about the results. And later, Spemann would win the Nobel Prize primarily for this work. But the Nobel Prize Committee, they never give out prizes posthumously, so she got nothing. By the way, her son Christian, Victor Hamburger says that he was killed in what Victor calls Hitler's war. So she has no descendants among us. Okay. So in 1935, or whenever Spemann won the Nobel Prize, this organized experiment was clearly the most famous experiment in biology, certainly in embryology, certainly in developmental biology. And so everyone was very excited that all we have to do is figure out what the dorsal lip of the blastopore is doing to kickstart a new organism. And as what would happen in the years after is nothing. So it used to be a joke that the organizer experiment had brilliantly impeded developmental biology progress for 40 years because nobody could figure out what to do with it. And people started to doubt it. Well, maybe all that happened is she just killed some cells in this organism, and somehow it was the killing of those cells that made them behave abnormally, and that's why another one developed, right? But it wouldn't be till well after the middle of the 20th century that the organizer concept was vindicated, and involved experiments in Xenopus, African clawed frogs, where people had been, studying development in frogs for a long time. It's great, right? The zygote's out there in fluid, and you can manipulate it, and they're pretty big and stuff like that. And people had noticed something. One of the things they noticed is that if you add a little lithium chloride to the embryos, they make these enormous heads. It's like, well, that's not right. And if you put in too much lithium chloride, the organism wouldn't be viable. It could be nothing but head. And they also noticed that you could do something else to an embryo if instead of lithium chloride, you radiated it, you could make embryos that would fail to make a head, fail to make an organism at all. So they took advantage of this. So here is a UV irradiated embryo, no head forms. And so what they did was they took embryos that had been exposed to lithium chloride that would normally make a bunch of head, and they extracted the mRNA there and injected various fractions of mRNA into a UV irradiated embryo, one where they knew normally no organism would develop. Everyone got that? because they'd worked out the parameters of how to irradiate them. And lo and behold, with some fractions of the mRNA, they could rescue the animal. They could rescue the phenotype. They could end up with a normal tadpole. And so that was very encouraging. There's one of these mRNAs must be important as the organizer. If you irradiate the embryo, you kill it. But if you extract mRNA from an embryo that's been jazzed up to have lots of organizer stuff, then you can rescue it. So then they started making a cDNA library and fractionating, finding various mRNAs to try to see which one would work. So if they injected nothing in irradiated embryos, you got nothing. Most mRNAs did not rescue the phenotype, but there was one gene injecting that mRNA into the organism would cause it to develop. Again, they rescued the phenotype. They overcame the effects of the irradiation. And so they reasoned that Noggin, and they named Richard Harlan did these experiments, and he's British, and so the British like to call heads Noggin, so he named it Noggin. And what he found is there was a dose-response curve. If you provided no Noggin to these irradiated embryos, you got nothing. But if you increase the dose, you could get normal embryos. And if you increase the dose too much, you got embryos that were all head. So Noggin was definitely inducing the formation of a head. And once they sequenced the gene and did in situ hybridization to see where that gene is being expressed, lo and behold, it's being expressed in the dorsal lip of the blastopore. How's that? So there is an organizer, and it's called Noggin. Good. Allison?
We know now, obviously, that the dorsal lip of the blastopore is like the organizer. Did Spemann and Hilda ever try any other part?
They did, and so taking any other portion of a donor embryo and engrafting it in, you got nothing. You got ordinary regulation. If you didn't mess things up too much, the organism would develop, but it looked like just any other. Well, it soon turned out that the field had an abundance of riches because eventually people would, using the same strategy, would find two other organizers. So there are 3 different genes, any one of which can act as an organizer. And their names are Noggin, chordin, and follistatin. And it looks like there's some redundancy in this system. So when they finally made, and this was back in the old days when it was hard to make knockout mice, not like today, when they finally knocked out Noggin in mouse embryos, there wasn't much of an effect. Maybe a little bit, there's an earpiece missing there, but it's like, wow, there must be another organizer. And indeed there is. So if you made a double knockout, where you knock out both Noggin and chordin, now you get no nervous system. And as I said, eventually people would find another gene that can also serve as an organizer called follistatin. How are you doing so far? And here I was going to talk about how you make knockouts in the old days and but you've run into that before. I'll just say that all of these manipulations, when they work, it's because you end up with animals where, no, I don't, it's not, it isn't, yeah, never mind. You've heard all that before. Yeah. Anam. Yeah.
So when you knock out Noggin, you still get like a pretty functional.
Yeah. So, right. No, it just means that you kind of have to knock out at least two of them to get a real prominent phenotype.
So if you knocked out chordin and follistatin, you can
Probably someone did that, but I don't remember. But yeah, I mean yeah. Once the genes were identified, you can be sure people tried all sorts of manipulations, and this is a theme that we'll see more and more often, especially in mammals and other vertebrates, is there's often redundancy in these systems, right? Yeah.
So do all three of these genes have like the same exact function in the way they organize, or do they go about it?
What a great question. Let's ask, how do they work? And indeed, they all work through the same mechanism. To understand this, first of all, let's talk about what the dorsal lip of the blastopore does. It invaginates, as we said before, and eventually these cells are going to make the mesoderm, which is just lying underneath the ectoderm, because the blastocele is getting displaced entirely, and there's the endoderm. Okay? So that initial set of cells that invaginate, they're the beginning of the mesoderm. And so you end up with a situation where there's a line of mesoderm that is directly under ectoderm. And I'm not going to go through all the experiments that showed this, but it turns out that all three of those organizers are being expressed in the dorsal lip of the blastopore. And they are all working because they interfere with binding of bone morphogenetic protein. Huh? Okay. So, it turns out that the ectoderm is constantly releasing the secreted factor that is known as bone morphogenetic protein, or BMP. And there's a lesson there already. Can you guess why it's called bone morphogenetic protein? Well, that's because the first people who found and isolated it, and therefore had the honor of naming it, I'll bet they were studying bones, right? But it turns out it's not just important for bones. This factor is important in the very beginning of embryology, right? It's crucial. And we'll run into this again and again, where we will see the names of genes, I'm sorry, offer you no clue as to what role it's playing in the story we're telling. Because the same gene, natural selection, does not do things the way an engineer would, right? The same genes, especially signaling proteins, they get used over and over again. Over and over again, natural selection hits upon using them to get some task done. So bone morphogenetic protein, it normally binds to an extracellular receptor that's known as TGF-beta receptor, transforming growth factor beta receptor. Oh, why is it called that? Because the people who named that gene were studying cancer, where transformation of cells refers to cells kicking out of their normal phenotype and becoming rapidly dividing cancerous cells. You can watch that in Cells in a Dish, and it's called, they're said to transform. Okay. So now it's transforming growth factor, which you'd think from the name must be important for cancer, and it is. But its normal ligand is bone morphogenetic protein. So just get used to it. There is another lesson here, though, and it's one that I don't think gets talked about often enough. If you're going to make a really complicated organism and you've only got 20,000 genes, you're going to have to make every gene do a lot of different jobs. And there's a term for that, when a given gene is involved in several different functions, and it's called pleiotropy. And I would say, especially for signaling proteins, pleiotropy is not the exception, it's absolutely the rule. It's almost universal. If it's a signaling protein where one cell can talk to another, natural selection will have incorporated it into several different places, several different times. And this is why there'll be situations, and we'll run into some in chapter 3, where the loss of a single gene can result in someone who can't smell and is deaf. In the early days, why is that? I mean, how can the same cell be important, same gene, be important for both smelling and hearing? Well, because the gene exhibits pleiotropy. It's vital for several different systems. And that's the rule, not the exception. Okay, so in the blastula, ectodermal cells are secreting bone morphogenetic protein all the time. And you can think of bone morphogenetic protein as being a unifying system. We're all epithelia, and we're going to stay that way. I'm epithelia, here's some of my bone morphogenetic protein. You're epithelia, I'll take some of yours, and we'll just keep talking to each other, and by golly, we're going to be epithelia. But the organizer interferes with this. So the organizer is an endogenous antagonist to bone morphogenetic protein. So all three of them interfere with the binding of the TGF-beta receptor, and therefore the cell no longer getting the signal, hey, we're all epithelium and we're going to stick together. The cell says, oh, I'm going to be a neuron. How's that? And the signal, the organizing signal, is being secreted by the mesoderm, and there's just that one line of mesoderm in the midline, and it is telling the cells above it, hey, you're being called for a different function. You're not going to be skin, you're going to be the central nervous system. And so once the neural tube forms, the source of the bone morphogenetic protein, the source of the organizer, is this structure known as a notochord, a rod-like structure right down the middle of the organism. It's made of mesoderm, and it's secreting signals. And so what's going to happen is once the neural tube forms, well, the mesoderm induces those cells to become the neural tube. They abandon an epidermal fate and take on a neural fate and invaginate and form the tube. And now we've got the neural tube that's getting the neural signal from the notochord, and the cells over the neural tube, the signal's too weak out there. And so now they go back to swapping bone morphogenetic protein back and forth between each other. We're bone morphogenetic protein, right? It's a cheer. Everyone ready? BMP, BMP. They're all sending it out, and they're all responding to it. And by golly, they're going to be unified in their fate. They're going to become skin cells. And these cells would have become skin cells, but the organizer interfered with the signaling, and therefore they took a different pathway. So none of you have asked yet, who arranged for the organizer to be in the dorsal lip of the blastopore? And I already foreshadowed. I mean, eventually the answer is mom, right? But we know a little bit about that process. So it turns out in the dorsal lip of the blastopore, as it forms, there's a set of cells down here that are secreting a protein called Beta-Catenin. Don't ask why it's called that. You just got to live with it. That's what it's called. And the Beta-Catenin, below the dorsal lip of the blastopore, induces the cells above it to start secreting organizer. And as invagination continues, thought I had one more. And as invagination continues, they will make the mesoderm underneath the ectoderm and induce the overlying ectoderm to make the nervous system. And of course, who put the Beta-Catenin there? Mom. And if you make mice that overexpress Beta-Catenin, and therefore they make more organizer, and therefore you end up with these brainiac mice that can't survive at birth, right? Because they've got so much brain in there that they can't get through the vaginal canal. How's that? Good time for questions. Okay. So I'm going to wrap things up, but when we come back, we will finish chapter 1 and start chapter 2, and I'm going to present you with a theory about why invertebrates have their nervous system on their bellies and vertebrates have their nervous system on their backs, that I promise, if you haven't heard it already, is going to blow your mind. So, see you Thursday.